Welcome to the lecture on sensation and sensory receptors. I am Professor Arbinderjeet Singh from the Faculty of Medicine, University Technology, Nara. Now, what I hope to do in this session is to talk about the different types of somatic sensations felt by the body. I shall not talk about the special senses, in other words, the eyes and the ears and the taste. All right? So I'll just talk about the somatic sensations felt by the body. I'll also talk about the types and characteristics of peripheral sensory receptors. In addition, I will talk about the mechanism of action of these receptors, in other words, how they detect the stimulus. And I will very briefly also talk about adaptation in these receptors. I'll talk about the stimulus strength relationship to the receptor general potential, and also to the frequency of firing in the nerve. And I'll very briefly talk about the receptive fields. Yes, so this is what I hope to cover in this lecture. Now, the nervous system can be divided into three main functional divisions, as you will know. You have the sensory division, and this division is actually responsible for detecting changes in the internal and external environments, and it informs the central nervous system about these changes. It is responsible for the perception of the sensations, which is what we'll be talking about. So that's the sensory division. You have the motor division. This division is responsible for initiating and controlling movement and activities of skeletal muscles. It also controls the activities of the other muscles, the striated, the smooth, the cardiac muscle, and even the activity in some of the glands. And then you have the third division, the intellectual or cognitive division, that is responsible for consciousness, memory, learning, thoughts, and emotions. So roughly this is how the three main functional divisions of the nervous system are. But in this lecture, I will talk mainly about the sensory functions of the nervous system. What is sensation? Sensation is a conscious perception of a particular feeling caused by stimulation of a certain type of receptor by its adequate stimulus. What do we mean by adequate stimulus? Adequate stimulus usually refers to the type of energy to which a sensory receptor responds to with the initiation of sensory transduction. All right? So each receptor will have its own type of stimulus. All right? So we call that the adequate stimulus. So are there different types of sensations felt by the body? And if they are, what are these sensations? Very broadly, the types of sensations in our body can be visual, which is sight, auditory, hearing, gustatory, taste, olfactory, that is not small but smell. Yeah. You also have the somatosensory sensations. These include the touch, pressure, vibration, temperature, pain. This is what I will be concentrating on in this lecture. And then you also have the osmoreception. These detect changes in osmotic pressure, osmolality of body fluids. And you have chemoreception as well. That's another type of sensation. And these normally detect changes in gas composition of the blood. Now, the sensory input to the nervous system is provided by the sensory receptors. Okay. And these receptors are the ones that detect the sensory stimuli. It could be light, sound, pain, touch, cold, warm, pressure, whatever it is. These receptors pick it up, they sense it, and then they send the input to the nervous system. And information, of course, is relayed to the brain through specific pathways which form the sensory system. I will talk about these pathways in the next lecture. So in this lecture, I will primarily concentrate on the sensory receptors. So what are the components of the sensory system? So before I talk about the receptors, let's quickly look at the components of the sensory system. This diagram here picture just depicts very broadly the main components of the sensory system. Of course, it starts off with a receptor. All right, the receptor is then connected to a nerve, a neuron, and that becomes the first order neuron. All right, so it carries impulses from the receptor into the spinal cord. So that's the first order neuron. So this is the afferent nerve. All right, and in the spinal cord, it will synapse with another set of neurons, and it might cross over, and then it is carried all the way up to the thalamus. And these would be the second order neurons. So you have the first order neurons from the receptor to the spinal cord. The second order neurons start from the spinal cord right to the thalamus. That's how the sensation is relayed. It doesn't matter what the sensation is, whether it's pain, warm, temperature, cold, or whatever it is. This is approximately what happens. These are the components. So you have the receptor, you have the afferent neuron, which is the first order neuron. It synapses with the second order neurons in the spinal cord, and the second order neurons then go all the way up to the thalamus, where they synapse with the third order neurons. So these third order neurons carry that information right to the primary somatosensory area of the cerebral cortex, and that's where the information is interpreted and the site is identified. So very briefly, the major components of the sensory system consists of the receptors, the afferents. All right, so the afferents we talk about, the first order neurons, and then the tracts we talk about, for example, sensations of pain and all these are carried by the second order neurons in the lateral spinothalamic tract. All right, and it goes right up to the thalamus. All right, from the thalamus, of course, you have the third order neurons, and these neurons transmit information right to the primary somatosensory area of the cerebral cortex. So this is approximately what the sensory system is and the components of the sensory system. And we'll talk about these in the next lecture as well. How many types of receptors are there? Because we have various different types of sensations. So it would mean that there are many different types of receptors. Now, the sensory receptors that provide input to the brain can be divided into 5 main types. You have the Mechanoreceptors, and these are usually excited by some mechanical pressure, mechanical deformation. All right, so these are Mechanoreceptors. Then you have the Thermoreceptors. These are usually excited by changes in temperature. You have Nociceptors. These are excited by noxious stimuli like pain or damage to the tissues. And then you have the Chemoreceptors. All right, these are the ones that detect taste, smell, or this detect gas compositions in your body, osmolality. All right, and a few other substances that you have in your body. And you also have the electromagnetic ones. These are usually excited by light. So very broadly speaking, the various types of receptors in our body can be divided into 5 major types, right? You have the Mechanoreceptors, Thermoreceptors, Nociceptors, Chemoreceptors, and electromagnetic receptors. So how do 2 types of receptors detect different types of sensory stimuli? That you might ask. All right? You may ask, how do these receptors differentiate different types of sensory stimuli? They do that through differential sensitivity. And what do we mean by differential sensitivity? Is that each type of receptor is sensitive to one type of stimulus generally. Okay, so each type of receptor is sensitive to one type of stimulus. How do we experience these different modalities of sensation? As nerve impulses are all conducted in the form of action potentials. How can we tell the difference? How do we tell where or which part of the body it is coming from? Each nerve receptor has its own specific nerve fiber. And this nerve fiber transmits information to the brain, and these nerve tracts terminate at a specific point in the brain. So the type of sensation felt is determined by the point in the brain at which the fiber ends or leads to. That is how we are able to tell. Right? Yes, all the impulses are conducted in the form of action potentials. But how are we able to tell what it is and where it is? It is by where it ends in the end, where the nerve tract terminates in the specific point in the brain. Approximately that's how we are able to differentiate. The specificity of nerve fibers for transmitting only one modality of sensation is known as the labeled line principle. All right. So as I mentioned just now, each nerve receptor has its own specific nerve fiber that transmits. All right. The nerve receptor or a group of receptors, if they have their own nerve fiber that transmits information, and that is a labeled line principle. All right. That is what we call the labeled line principle. Or sometimes it's also law of projection. So no matter, and this is an interesting phenomenon, because no matter where along the nerve pathway one stimulates, the sensation will be felt at the site of the receptor. All right. So what it means basically, if you have a receptor and you have the nerve that is transmitting information from that receptor to the brain. Now, if that particular nerve is stimulated anywhere along its path right to the brain, the impression the brain will interpret it as if it's coming from that particular site. So that is what we mean by law of projection. All right, so we'll talk about some sensory receptors, and for this we will talk about the receptors in the skin. All right. So what types of sensory receptors are there in the skin? You have many different types of sensory receptors. You have the free nerve endings. All right. These are the ones that are sensitive to pain, heat, cold. Then you have the Merkel Discs. These are sensitive to touch. You have the cross-end bulbs. These are also sensitive to touch. You have root hair plexus. All right. These are also sensitive to touch. Then you have the Meissner's corpuscles. Again, these are also sensitive to touch. Then you have the Pacinian corpuscles, which are sensitive to pressure. All right. You also have the Ruffini endings, which are also sensitive to pressure. So as you can see, there are various different types of receptors present in the skin. Of course, you also have the sensory receptors that are found in the muscle and the joints. You have sensory receptors in the muscle and joints, muscle spindle, the Golgi tendon apparatus. Yeah. So these are the various different sensory receptors that are found in the skin. How do they detect the stimulus? The adequate stimulus. How do they detect it? In other words, what is the mechanism of action of these receptors? Okay, the mechanism of receptor potential. Now, these receptors can be excited in one of several ways, as you would have realized by now. You have the touch receptors, the pressure receptors. These perhaps will respond to mechanical deformation. All right, so there is mechanical deformation. You have change in temperature. So these will be sensed by the temperature receptors, all right, thermoreceptors. Chemical receptors will sense chemicals, all right. And you have the electromagnetic radiation like light and all that will be detected by the electromagnetic receptors. All right? So these receptors can be excited in various different ways, and of course each receptor has its own way of being how it gets excited. All sensory receptors respond to a change in membrane potential when stimulated. So it doesn't matter what the stimulus is, which receptor it is. Once the adequate stimulus is applied, that particular receptor will respond by changing its membrane potential. There is a change in membrane permeability, and there will be a change in membrane potential. And this change in membrane potential is known as the receptor potential. So in other words, when a receptor is stimulated, it generates what is known as the receptor potential. And in some receptors, the magnitude of the receptor potential can be as high as or as much as 100 millivolts. Now this receptor potential that is generated in the receptor then stimulates the generation of action potential in the attached nerve. So as you have learned earlier, as I mentioned earlier, each receptor is connected to a nerve, all right? And when the receptor potential is generated, it will then stimulate the generation of action potential in the attached nerve. How is receptor potential generated, you may ask? How is it generated? All right, we'll look at an example, for example, in the Pacinian corpuscle. And you will see that in a lot of physiology textbooks, and I've taken this from Guyton. And it very briefly explains what happens when pressure is applied. So you have a capsule, all right? So this is actually placed in a capsule, for example. That's why it's called a corpuscle-like, right? And when pressure is applied to the capsule, all right, so inside this Pacinian corpuscle, you have concentric layers, capsule layers, all right? And what happens when you apply the pressure, these layers will get indented, they will elongate, or they get deformed, all right? So when pressure is applied, you are actually disturbing the formation of these layers, if you like, all right? So they get a little deformed. And that deformity that takes place, so this, when it happens, what it does is opens up the ion channels at the region of where the deformation is. And when the permeability of that part of the membrane changes, the ion channels, of course, open. That is a cause of the change in permeability. And there is entry of positive ions into the cell, the receptor. And that creates positivity inside the receptor. All right? So this is, in other words, the receptor has altered its membrane potential. As you can see, this is what has happened. There is depolarization, if you like. Because it's in the receptor, we call it the receptor potential. All right? Now this receptor potential will then give rise to action potentials in the nerve that is inside the receptor itself. So in this instance, in the Pacinian corpuscle, the nerve actually is embedded within the corpuscle. All right? So the change in the membrane potential in the receptor will then cause a change in the nerve that is lying within the receptor. All right? We call this the generator potential as well. So the receptor potential that is generated will induce local circuit currents or current flow that spreads to the nerve fiber at the first node of Renvier. And as I mentioned just now, usually the first node lies within the capsule. And then this then sets off the action potential in the whole nerve. All right. So once the action potential is triggered in the nerve, it is then transmitted along the nerve all the way right to the brain. So this is how the receptor potential is generated. So if you are asked to explain how the receptor potential is generated, you could use the Pacinian corpuscle as an example. All right? And what happens here, if I may repeat, when you apply pressure, when the pressure is applied, that changes the membrane permeability. All right? The deformation that occurs when the pressure is applied changes the membrane permeability. The ion channels open, there is entry of sodium or positive ions into the corpuscle. All right? And this, of course, will change the potential of the receptor. All right? So it depolarizes. And when that part of the receptor is depolarized, it sets up circuit currents within the nerve itself. So when these circuit currents, then the current starts to flow. All right? And as it spreads to the nerve fiber, it reaches the first node of Renvier. All right? And then from there, it of course goes from node to node. All right? And so once it gets into the nerve, it generates the action potential in the nerve. All right, so this action potential then reaches the nodes of Renvier, the first node of Renvier. From there, then it is transmitted by saltatory conduction along the nerve. All right. Now, you may ask, does the amount of pressure applied affect the receptor potential? The answer is yes. All right. The more the pressure, the greater the receptor potential. So the amplitude of the receptor potential increases as the stimulus strength is increased. And this picture depicts that. As you can see, as the stimulus strength increases from 0 to 100, you can see the amplitude of the receptor potential. It also increases. Of course, it plateaus later on at higher stimulus strength. But nevertheless, the relationship is that it increases with the stimulus strength. It increases rapidly as first, as you can see, but then less rapidly, all right? So when the receptor potential rises above the threshold for eliciting an action potential in the nerve fiber, action potentials begin to appear, all right? And the more the receptor potential rises above the threshold, the greater the frequency of the action potential, all right? And this diagram or picture depicts that. As you find, as you increase the, as the stimulus strength is increased, and you can see what happens to the frequency of firing of the action potentials in the nerve. So that's how our brain is able to tell how much pressure is applied, for example. More the pressure, higher the frequency of this action potential reach the brain, and the brain will then interpret it as higher pressure. All right? So this is what happens. So basically there is a relationship between stimulus strength and receptor potential and the frequency of action potentials that are generated in the nerve. Can the receptors be stimulated from anywhere? Do they have a specific area? All right, that's the next question you might ask. So far we have spoken about the different types of receptors. We have very briefly spoken about the mechanism of how these receptors are excited, and we have spoken about the adequate stimulus, that each receptor has its own specific stimulus that stimulates it. And we have also learned about the relationship between stimulus strength and the receptor potential. All right, so we have learned that as the stimulus strength increases, the receptor potential increases, and with it also the frequency of action potentials that are generated in the nerve. So the next thing you may want to know is, can the receptors be stimulated from anywhere? Do they have a specific area? The answer is yes. Each receptor has its own specific area, which we call the receptive field. Right? Now, a receptive field of a sensory neuron is a region of the sensory area in which a stimulus will modify the firing of that neuron. In very simple terms, it is the region where, within the region, when you apply a stimulus, it will generate receptor potential. That's what it means. If the adequate stimulus is out of the receptive area or receptive field, it will not elicit a receptor potential. Okay, so each receptor has its own receptive field within which it will be able to sense a change in pressure or whatever the stimulus is. And this diagram captures what I've just mentioned just now. If you can see the receptors, and you can see their receptive field. All right, so you have the receptive field 1 is for this receptor, receptive field 2, receptive field 3. All right, so an individual receptor will be activated by stimuli that falls within a specific area, within its own receptive field. All right, but this receptive field, the size varies depending upon the dendritic abo of the individual receptor and the position of the receptor in the sensory organ. All right, it can vary. So the receptive field for receptors can vary. And so you might ask, where would you find small receptive fields and where would you find large fields? So in some parts of your body, where, for example, the sensitivity is not required to be very high, you might find a slightly larger receptive field, all right, and less receptors. In areas where you have to be very sensitive, you probably will find many receptors, smaller receptive fields, but many receptors. And these receptors might also be actually leading to more than one nerve as well, all right? And we'll talk about this shortly. We'll talk, now I'm going to talk about the purpose of the receptive field. What is the purpose of the receptive field? Now, it's through the receptive field that you can actually discriminate in the 2 points of stimulation, for example. All right. Now, a small receptive field, they permit high resolution of spatial detail. In other words, they can discriminate between 2 points of stimulation. All right, you can see here in this instance, the picture here below, that will show the sensory receptors, and you have a divider here. All right, and you have the 2 receptor fields. All right, and look at the second one where you have a receptor field and the divider is within that same field. So it, although there are 2 points that are being stimulated, but it will be perceived as just one. On the left, you see a diagram where there are 2 circles with a divider, arm in each circle. Here you find, because of the 2 receptive fields, it will be interpreted as 2 stimuli. So there is a discrimination. All right. So this is the significance of the receptor fields. All right. Now, there are 2 ways, of course, we can discriminate. All right. One, of course, is having the receptive field. All right. So you find, for example, here in the picture, figure A, the many primary sensory neurons, they converge onto a single secondary neuron. So even if you have 2 separate receptive fields, but if they converge onto a single secondary sensory neuron, it will go to the brain as just one signal, and it will not be able to discriminate these 2 points. But if you look on your right here, you have these receptors, all right, and when you use the divider and you touch these 2 receptor fields, because they are separated and they are going to separate secondary sensory neurons, it will be sent to the brain as 2 signals. All right. So when fewer primary neurons converge, secondary receptive fields are much smaller. All right. So the 2 stimuli activate separate pathways and are perceived as distinct stimuli. These are some examples. You may probably do that in the practical as well. You look at the tongue. If you have these 2 points of the divider, if they are placed on the tongue less than 2 millimeters apart, the tongue will just perceive it as one signal, all right, as 1 point. Similarly, at the fingertip, for example, if it is less than 4 millimeters, it will be perceived as 1. But if it is more than 4 millimeters, the distance between the 2 points, it will be perceived as 2. And look at the shoulder, for example. If it is less than 64 millimeters, all right, it will be perceived as just one signal at 1 point. But if it is more than 64, it will be perceived as 2. So you find, look at the sensitivity, where you have the highest sensitivity, of course, the tongue and the finger. All right. So because the shoulder is, it's not so important to discriminate, or the need for such fine discrimination. Yeah. Now, the other mechanism, of course, that helps also in the discrimination, apart from the receptor fields, all right, the location of the receptor field and how they actually, the impulses from the receptor field, how they are transmitted to the neuron. If it is just a single sensory neuron, it will be just one signal. If it is 2, it will be interpreted as 2 signals. So that's one way of having this 2-point discrimination. The other thing that helps in the discrimination is also lateral inhibition. Now, lateral inhibition is the phenomena in which a neuron's response to a stimulus is inhibited by the excitation of a neighboring neuron. All right? So when you have a very simple analogies, of course, if you have a class full of people and all of them are talking at the same time, you won't be able to hear. All right. So what you have to do is you have to ask everyone to keep quiet, and then when one person speaks, you hear that person very clearly. So this is exactly what is happening. So what do they do? Some receptors, they want to make sure that their signals are conducted right to the brain, and they don't want the information that they are going to conduct to be disturbed by the neighboring sensory receptors. They actually inhibit those receptors momentarily. all right? And so only the signal from that receptor gets transmitted to the brain. So we call that lateral inhibition, all right? So in short, the lateral inhibition will make the neurons more sensitive, all right? I mean, this particular feature or characteristic is used in a number of areas of the brain as well. And even in the retina, for example, the neighboring cells can inhibit each other, so that a tiny point of light on one cell will tend to stand out in contrast to the adjacent ones. In touch, for example, neighboring cells in the skin also use lateral inhibition. So as I mentioned just now, the functions of lateral inhibition is to basically able to discriminate better. It enhances the contrast, so it's for contrast enhancement and also maximum selection. Now, do the receptors continue to fire at the same rate when stimulated continuously? So as we have spoken just now, when you stimulate the receptor, when the pressure is applied or the adequate stimulus is applied, receptor potential is generated and action potential is generated in the nerve. The question that you will ask next is, do they continue to fire at the same rate all the way through, as long as the stimulus is there? The answer is no. All right. Some receptors only fire for a short while and then they stop firing. Others will probably fire for a longer period, but the rate of firing becomes less. All right. And some of them will continue to fire for a very, very long time. All right. Now, we call this process, those that actually stop firing, we call it adaptation. All right. So we know that all sensory receptors adapt. Some adapt very quickly, some adapt slowly, some adapt partially, and some adapt completely to the constant application of stimulus. All right? But if you take away the stimulus and then apply again, of course they will be sensitive. But if you keep the stimulus applied continuously, this is what happens. The receptors adapt. All right? So the receptor at first responds at a very high impulse rate, and then progressively it responds at a slower rate until, of course, the rate of the action potential firing decreases to very, very few, all right, or even to none at all. All right? It stops firing completely. Now, as I mentioned just now, some receptors, they adapt faster than others. All right. Here you will see that examples of some receptors in this diagram. All right. Some adapt very rapidly. Mechanoreceptors adapt almost completely. Non-mechanoreceptors like the chemoreceptors, pain receptors, they never really adapt completely. So you can see here the different adaptation rates of the various receptors. So you have the Pacinian corpuscle, you find it is a very rapidly adapting receptor. The hair receptors is the next one. All right, within a few seconds it adapts. The muscle spindle, for example, all right, it partially adapts. It doesn't adapt completely, of course. And then you have the joint corpuscle receptors as well. They too partially adapt, not completely. So in short, do all these receptors adapt? We say yes, receptors eventually adapt to the if the stimulus is applied continuously to the receptor's response will gradually decrease. And we call that adaptation. All right. And some receptors adapt completely. In other words, despite the stimulus still being there, they stop firing. Others will perhaps decrease the rate of firing. All right. So you have partial adaptation. How do they adapt? So if they're adapting, what really happens? How do they adapt? The mechanism of adaptation. Still look at the example. We can use an example, the Pacinian corpuscle, for example. Now, there are two major mechanisms of adaptation. All right, you have readjustment of the structure of the receptor itself. All right, or it could be an electrical type of accommodation. All right. So these are the two major mechanisms that are involved in adaptation. Like readjustments to the structure, for example, if you look at the Pacinian corpuscle, when you apply pressure on the Pacinian corpuscle, there is an indent, there is a deformation takes place, all right? And this indentation or the deformation that has taken place, all right, is still there if you apply the pressure, and that will change the membrane potential and the receptor potential is generated. But if you keep the deformation there, what happens is that, you know, there is fluid inside the corpuscle, and this fluid redistributes itself. Consequently, the receptor does not feel the deformation or the distortion in the layers, and it stops firing. All right. So this is one mechanism. In other words, there is a readjustment of the structure in the receptor. That explains the adaptation. The other one, of course, is this progressive inactivation of sodium channels in the terminal nerve fiber membranes. All right. So when there is inactivation, this will also result in adaptation. So these are the two major mechanisms that have been used to explain adaptation in receptors. Now, do they adapt at the same rate? Of course, the answer is not. As I mentioned earlier, some are rapidly adapting, some adapt rather slowly. All right. So you have the rapidly adapting receptors. These receptors initially will react very strongly when the change is taking place. All right, and then gradually they adapt. All right. So in other words, they only respond to the change in stimulus strength. If the stimulus strength does not change, if you keep the same strength, they will not respond. They stop responding. And then there's a change in stimulus strength, they will respond. We call these phasic receptors. All right. And the disadvantage of these receptors, of course, if you want to transmit signals continuously, these receptors will not do. All right. So because they stop getting stimulated after a while. So they cannot be used to transmit continuous signals. Example is, for example, the Pacinian corpuscle we've discussed just now. So when you apply pressure, a sudden pressure is applied to the tissue, it excites the receptor for a few milliseconds. It transmits the signal, all right, for a while, and then it stops. But if you were to release the pressure and apply again, then it will transmit again, all right? Or when the pressure is released, it will transmit the signal. So it responds to the change in pressure. So it's a very rapidly adapting receptor. You have the slow adapting receptors, and these detect continuous stimulus strength. All right. So the rapidly adapting receptors normally detect while the change is taking place. The slow adapting receptors will detect continuous stimulus strength. All right. So they are known as the tonic receptors. They continue to transmit impulses to the brain as long as the stimulus is present. It could be hours, days. They keep the brain constantly informed of the status of the body and its relation to its surroundings. And examples of these receptors, the slow adapting receptors, include the Golgi tendon apparatuses, all right, the macular and the vestibular apparatus, for example, the baroreceptors, the chemoreceptors. These are some of the examples of the slow adapting receptors. All right. Now, I hope I've covered most of what I had planned to do. So in this lecture, what I had planned to do was, of course, to introduce to you the various types of sensations and also the types of receptors and their adequate stimulus, and very briefly talk about the mechanism of generation of receptor potential, and we use the Pacinian corpuscle to illustrate the mechanism. And then we spoke a little bit about some of the characteristics or features or properties of these receptors. We spoke about the stimulus strength and the receptor potential relationship. We talked about the stimulus strength, receptor potential, and the frequency of firing, the relationship. And we also spoke a little bit about adaptation and what happens in adaptation, the mechanism of adaptation. Right? So the next question will be, what happens to these signals from the receptors? So receptors have generated the signals. What happens to them? How are they transmitted to the brain and interpreted? All right. And this will be continued in the next lecture. Thank you very much.